nervous system.
Oh, okay.
Okay, I think it's recording now. Okay,
Alright.
so the first question is here. On the male reproductive system, so a 24-year-old man presents to the fertility clinic with concerns about not being able to father a child after 2 years of unprotected intercourse. He has no significant medical history, and he denies any use of tobacco, alcohol, or any recreational drugs. So he's not on any medication. Examination shows he has a normal secondary sexual characteristics, no abnormalities in the genital examination. Semen analysis reveals low sperm count or oligospemia, and also poor motility. Blood tests show normal testosterone levels, but there is elevated follicle stimulating hormone. Scrotal ultrasound shows normal testicular size and no varicella. So in this case, this gentleman, everything else seems to be normal, but then he just has a low sperm count. Okay. And then also they did an ultrasound on the scrotum, and they discovered the testicles are just normal size. They don't even have any fluid in them, so everything looks to be normal. Okay. So the only issue is here. So there is low sperm count, and again the type of spermatozoa being produced, they have poor motility, meaning um, they are not moving. Okay, so there could be a number of problems that can be as a result of, first of all, poor motility, okay, and also a low sperm count. So there could be a number of problems that can cause these two. So the first thing here, for poor motility, maybe just to explain, but this one also, it's under histology. If there is a problem in cilia, there is a certain sometimes microorganisms that attack cilia growth, and type of cilia that forms the male cell, it's called flagella. It's the one that forms the sperm tail. Yeah, so if somebody has got a challenge, these are called specialization. When you look at the epithelium cells, they get specialization, microvilli, cilia, and also what's that other type? Stereocilia. Yeah. So if there's a problem in formation of cilia, the flagella being formed is also going to have a problem, and there'll be poor motility. Okay, so that's just one of the things. But according to the examination, he doesn't look to have an issue with his organs. So what's the most likely site of dysfunction in this patient? Okay. So the most likely dysfunction here that can cause a low sperm count is the formation. Okay, I don't know if I've talked about this. You know the way spermatogenesis takes place. Okay, so having a low sperm count, it means so the semen analysis, you do a count. So you do a count. There's a way we do a count in the lab. At Ndola Teaching Hospital, they usually do semen analysis on Wednesdays. So you can
Every Wednesday.
yes, every Wednesday. That's when they do semen analysis. So
That's
what?
why you did your clinicals from?
Yes.
It was for how long? 1 month?
No, for I think that was 4 months.
Ooh.
So what you normally look at under semen analysis, you look at the size, the head, the tail, if they are moving, and then you do also a count. Okay, so if there is a low sperm count, it means that there's something that went wrong during spermatogenesis. Okay, spermatogenesis is just a process where it starts with a spermatogonia, it starts to split, then it forms like this, and then they develop tails. Okay, so this is what spermatogenesis is all about. So takes place. So the problem here, it's a dysfunction in the seminiferous tubules and also the Sertoli cell function, leading to impaired spermatogenesis. So spermatogenesis takes place in the seminiferous tubules. Now the Sertoli cells, these are not sperm cells. These are just cells that support sperm cells. So how do they support sperm cells? They help in providing nutrients. So they are just supporting cells. So if there is a problem here, meaning a lot of spermatozoa during formation, a lot of them die. I'm sure this is what is causing a low sperm count. And explain the physiological role of follicle stimulating hormone. So follicle stimulating hormone, this is a hormone that it is mostly talked about in females, but even males have. So follicle stimulating hormone, if it's in females, as the name suggests, follicles, it causes growth of follicles until the follicle grows into a Graafian follicle, and then it releases an egg. Okay. But in the male, the follicle stimulating cells is the one now that works for growth of these Sertoli cells. Okay. So in females, it works on the ovaries. So follicle stimulating hormone, it is produced straight from the pituitary gland, and then if it's in females, it will act on the ovaries. It will cause growth and maturation of follicles, which develop into an ovum, which is released. In males, FSH stimulates the Sertoli cells, and these Sertoli cells, they now give support. Okay, they give support to the growing spermatozoa. Okay, that's their function. And then also follicle stimulating hormone, it also promotes spermatogenesis. Okay, it also supports spermatogenesis. So that's the physiological role of follicle stimulating hormone in a male. So it will promote spermatogenesis and also stimulate Sertoli cells in the seminiferous tubules. Okay, so by supporting germ cell development and production of androgen binding protein. Then 3, name the cells that produce inhibin B and describe its feedback on the hypothalamic pituitary gonadal axis. Okay, so this is quite a long term. So maybe just to explain this so you can understand it. So from the hypothalamus, the way these things takes place is it's in the brain. In the brain, there will be the hypothalamus and the pituitary. So then if this is the head, and then now you have got the gonads now, which are the testes. So what happens is the hypothalamus, I'll just say H, and the pituitary, I'll just say P. Then these are the gonads, G. So this hypothalamic pituitary gonadal axis is where the hypothalamus secretes a hormone called gonadotrophin, and gonadotropin releasing hormone. It's called gonadotropin releasing hormone. Then it acts on the pituitary to secrete gonadotropin stimulatory hormone, which stimulates the gonads. Okay, so this is just the axis. So it comes from the hypothalamus, it goes to the pituitary, and then it goes to the gonads themselves. So now there's a hormone called inhibin. So the question says, name the cells that produce inhibin and describe the feedback effect on this axis. So the Sertoli cells produce this inhibin, and to exert a negative feedback on the anterior pituitary to suppress follicle secretion. So it's more like this. Okay, let me explain it like this so that you can just understand how it takes place. So when you have the hypothalamus, it will secrete gonadotropin releasing hormone, which will act on the pituitary, causing the pituitary to secrete follicle stimulating hormone. Follicle stimulatory hormone acts on the testis now. Okay, the testis, what does it do? It causes spermatogenesis, and also it causes stimulates the Sertoli cells. Okay, so now these same Sertoli cells, remember, they were stimulated as a result of follicle stimulating hormone. But after these Sertoli cells have been stimulated, they then secrete inhibin. So they will secrete inhibin, and after they secrete inhibin, inhibin travels back now to the pituitary to stop the secretion of follicle stimulating hormone. Okay, so that is it. So the cells that produce inhibin are the Sertoli cells, and once they produce the inhibin, it's just to suppress FSH secretion. But remember, FSH is the one that promotes or stimulates the Sertoli cells. That is why it is a negative feedback, meaning it's giving a negative feedback, meaning stop producing FSH. It's more like we are enough. The Sertoli cells are trying to say we are enough. We don't need further stimulation. Okay, we do not need further stimulation, so we don't need this. But again, you have to look at let's look at the question again. There's a problem here, because there was elevated FSH. So if FSH was elevated, it means that in this person probably the Sertoli cells had a problem. Okay, that is why here we said the dysfunction is most likely in the Sertoli cells. Okay, so now if the Sertoli cells have a problem, meaning they could not stop further secretion of FSH, that is why it was elevated. So the problem in this patient is a dysfunction of the Sertoli cells. Despite normal testosterone levels, this person had got normal testosterone levels. The patient has the reduced sperm count. Explain how this is possible. Okay, so let's look at what testosterone does. Okay, so testosterone, so this person has got normal levels of testosterone, but then he has a low sperm count, and we've said this low sperm count is attributed to the fact that there is no Sertoli cells. So the job of testosterone mainly in a human being, the job of testosterone is responsible for bringing about all secondary sexual characteristics, meaning all the characteristics that occur after puberty. Okay, once a boy reaches puberty, all those characteristics, deepening of the voice, enlargement of shoulders, and all those things, they are brought about by testosterone. Okay, and testosterone also stimulates the Leydig cells. Okay, but in this case, spermatogenesis requires mostly it is FSH and the functional Sertoli cells. So if Sertoli cells activity or the seminiferous tubules are impaired, sperm production is also reduced. So a person might even have a normal testosterone levels. Okay, so in this case, the only problem is these Sertoli cells, because they have to support the whole spermatogenesis function, and if they're impaired, means spermatogenesis did not take place properly. So describe the function of the Sertoli cells. Okay, like I've said, they are supporting cells, so they provide structural and nutritional support during spermatogenesis. So as the spermatozoa are growing, okay, so the Sertoli cells are there to provide the structural support and nutritional support to the germ cells. So they are very important in spermatogenesis. Okay, so they also so the Sertoli cells also they have a protective function. This is because they form a barrier. Okay, so what we call a blood testis barrier, it is a barrier that is formed by the Sertoli cells, preventing blood and the testis from meeting. Okay, so they also secrete androgen binding protein, they also secrete inhibin. Okay, so they simply, so the whole function here is just that they support protection. Okay, so they offer support to the spermatozoa. What type of support? There is structural, nutrition, and also protective support. That is why it becomes a problem if the Sertoli cells are damaged. Then what is the role of androgen binding protein? Remember, this is also secreted by the Sertoli cells. Okay, so the role of androgen binding protein, it is just a protein where testosterone can bind. So when you hear the word androgen, we are referring to testosterone. Okay, so testosterone can bind to this protein, okay, in the seminiferous tubules. So it is essential, okay, because if there are no Sertoli cells, it means that there will be no androgen binding protein, and no matter how much testosterone is produced, it's not going to cause any function. No, it's not going to cause any effect. Okay, so the whole case here, that's why the individual had normal testosterone. But just the fact that a person has normal testosterone, if the Sertoli cells are unavailable, also androgen binding protein will be unavailable. It means that testosterone will be there, but it's going to have nowhere to bind. And if it doesn't have anywhere to bind, it's not going to perform its function in spermatogenesis. Okay, so that is how it works. So for spermatogenesis, what is really important when it comes to endocrine? So for spermatogenesis to take place, what is really important is, first of all, follicle stimulating hormone. So the hypothalamus will first secrete the gonadotropin releasing hormone, then the pituitary will secrete FSH. FSH now goes to the testis, okay, and then the testis now, it stimulates the Sertoli cells. I'll just say CS. And the Sertoli cells now, they'll provide further functioning support, like we've said, they support. So this is where spermatogenesis now will take place nicely, if all these are available. Okay, one, gonadotropin releasing hormone should be there, follicle stimulating hormone should be there. There should also be presence of Sertoli cells, and the Sertoli cells, they will provide the ABP. The Sertoli cells also will provide support, and all these are factors that are going to be important in spermatogenesis. Okay, so in this particular question, the problem was here. Okay, the person had a problem in the Sertoli cells. That's why everything can be normal, but if this is not there, meaning this is not there, and spermatogenesis also be affected. Hence the person is going to have a low sperm count. Okay, this one just wants us to outline the stages of spermatogenesis. So spermatogenesis begins with a cell called spermatogonia. Then the spermatogonia now, it's a what can we say? It's a stem cell. Okay, meaning it is a cell that has a lot of potential to develop into many types of cells. So it's the spermatogonia. It is a normal cell having 46 chromosomes. Okay, so we are born with these spermatogonia, and especially in males. In males, this process it occurs throughout, okay, unlike the one in females. Females are born with a certain number of follicles, and but for males this is just a continuous process. So it begins with one cell, a very large cell called spermatogonia. So this spermatogonia will undergo mitotic division. So mitotic division is where this cell divides into two. Okay, so this is a spermatogonia. It divides into two, what we call primary spermatocytes. Okay, so the difference now is this. This one is going to have 46 chromosomes. This one is just a division, but it carries the same genetic information, and there are 46 as well. So when you say mitotic division, it means the same number of chromosomes are just passed on to the daughter cells. So that is only a primary spermatocytes. Then after this, now this is when the cell now goes into meiosis 1 and the meiosis. So these, they'll go into meiosis what? Meiosis 1. So under here this would go like this. Okay, so this is the enter meiosis 1. Okay, the enter meiosis 1. So after meiosis 1, now these 46 chromosomes are shared. Here 23, here 23. Okay, so 23, 23, 23, 23. So these are what? This is a spermatogonia. These are primary spermatocytes. These are secondary spermatocytes. So the difference between primary spermatocytes and secondary spermatocytes is that secondary spermatocytes have got half the number of chromosomes. Okay, because they undergo what? Meiosis, meiosis 1. Then meiosis 2 is just like this. There's no further reduction of what can I say? There's no further reduction of these. They're just given out just like that. They do not further reduce, so meaning they will just divide, but then they'll also have 23, 23, 23, 23. So now these are called spermatids. Okay, spermatids. And then these spermatids, now they undergo differentiation. That's when they start growing tails. Okay, so this is now, so the ones at the end now, these are now called spermatozoa, or the sperm cells. So these are now mature sperm. Then they start going through spermiogenesis. So now again you look at it, there's a difference. Spermatogenesis, it's all these stages. Then spermiogenesis is when this spermatozoa becomes into a fully adult sperm cell, because here once they are formed here, they have a lot of unnecessary materials which are cut off. Okay, so this is called spermiogenesis, where these now become mature. Okay, but from stage 1, 2, 3, 4 up to 5, it is spermatogenesis. Then spermiogenesis is just a process where they develop into mature cells. So now, for this process to occur, there are certain things that are needed. Okay, number one, like we said, there should be gonadotropin releasing hormone, which should cause the secretion of follicle stimulating hormone. Then there should be the Sertoli cells present. Also androgen binding protein, so that testosterone can bind to it. Okay, so all these should be available in production of normal sperm cells. So now you'll find that during the semen analysis, sometimes you'll find that there are sperm cells maybe with a very large head and a very short tail. So all this is an abnormality. Some you find that they are just having heads without a tail. Some you find that they are there but they are not moving. And sometimes you find that it's just semen without nothing at all, which is now a very even more difficult situation. But if everything is just there, they should form properly normal cells. Then number 8, describe one hormonal and one local intratesticular factor that influence spermatogenesis. So the hormonal factor that influence spermatogenesis, now they just said one, and here I just gave testosterone. But under hormonal, you can also give follicle-stimulating hormone. It influences spermatogenesis. So this, you can also give gonadotrophin releasing hormone. So gonadotrophin releasing hormone, follicle-stimulating hormone, testosterone, all these are hormonal factors. Okay. Then the local factors now. These local factors are those that have to do with the testes themselves. Okay. So such as Sertoli cells, they should be there, and these they provide a number of factors. So name the structure that is responsible for sperm maturation and storage. So the way a scrotum is, it's like this. Sorry, not a scrotum, a testis. And then the testis has got this structure around it here. Okay. So this is a testis. Then this structure here that I've drawn here, this structure, it is called the epididymis. Okay, it is called the epididymis. So the epididymis now, it is just there behind attached to the testis. So once the spermatozoa are formed, they are formed in millions. So they go into the epididymis now, and that is where they mature, and that is where they are stored right here. Okay, so they are made in very large numbers, then they are stored in this, and then so this is where now they are attached to the prostate. So the prostate comes in like this, then the urethra, and then that's how they lead to the outside. So the structure responsible is the epididymis. So the epididymis now, when you talk about histology, they have a certain, there are cells in the epididymis that have a specialization called the apical called the stereocilia. It helps. So remember, these spermatozoa here, they are just cells and they are stored here in numbers. So stereocilia here, these are cilia, they are found on cells in the epididymis. So they help to move these spermatozoa, because these spermatozoa, they need to move in a fluid, but here there's no fluid here. So they are helped by stereocilia. Then once they pass here, the prostate now, it's the one that secretes that semen, that whitish liquid. So that whitish liquid now, where the spermatozoa can swim nicely, then they come out in what we call semen. So from the prostate, that's where the semen is released, and it carries the spermatozoa. So now in complicated cases, you find that here there are no spermatozoa, but semen is released. So if you get this semen here, and you look through under a microscope, you find that there is actually no spermatozoa present, and so that's the worst case of infertility. Question 10, mention two components of seminal fluid. Okay, so seminal fluid, the same fluid that I'm talking about, that whitish fluid where the spermatozoa can swim. So it contains one, there's a sugar called fructose. Okay, so this fructose, it's what the spermatozoa feed on for energy. So the sperm cells, they need energy for them to move. And also there is a prostaglandin. So prostaglandin, it is a type of substance that causes contractions. Okay, it causes contractions. So so that white fluid that comes out, it has fructose. Fructose is what they use as their glucose, the spermatozoa. Remember, they are produced in many numbers, so they need fructose. So the way the structure of a spermatozoa, it has a head, and then after the head there is another part here called the acrosome, and then that's when then there is a tail. So this is the head, and the head is the only thing that combines with the ovum. But what is found here in the acrosome is that there is mitochondria. Okay, there is plenty of mitochondria, and mitochondria is known as the powerhouse because that is where energy is released. Now energy cannot be released without a sugar, so this is where the fructose comes from. So they use fructose to do respiration. So the prostaglandin, like I said, it's a substance that helps in contractions. So it helps in sperm motility, and if the semen is deposited in the woman, it's this same prostaglandin that can promote the uterine contractions to aid in sperm transport. So the job of prostaglandin, it's in the semen, and if the semen is deposited in the woman, that prostaglandin, it will help in contracting the womb so that this spermatozoa can further move upwards until they go into the fallopian tube, where they can meet a mature ovum. Okay, so two important things, fructose and prostaglandin. Okay, so we're done with this first question. So this one was just talking about spermatogenesis. Okay, and so the problem was just in the Sertoli cells. Maybe we can do the next question. Oh no, these are MCQs. I don't know if you're still there.